In my opinion, one of the most pressing questions in the field of multiple myeloma is how do we define lenalidomide refractory status? This is not an easy question. I think when we think about lenalidomide refractory patients, we have to define whether they're truly refractory. Now we can see that clinically where patients are getting, say, lenalidomide single agent and they progress. But sometimes the question comes up, for instance, in someone who's undergoing maintenance. Now, the more we have learned about the mechanism of action of lenalidomide, we know that patients may be sensitive or not more along the lines of a spectrum than in a very binomial fashion. Let me contrast this, for instance, with patients who have mutations in CML who may not respond at all to TKI. Imatinib will not work just because they have a specific mutation. I think myeloma is more gradual and there's a spectrum. Our audience knows that lenalidomide and other IMiDs work through the binding to this protein called cereblon, but the majority of patients who are deemed to be refractory to lenalidomide just don't have mutations or deletions of cereblon. So there's other mechanisms that are operating in how lenalidomide works. In our research laboratory, we have explored that, and there's some very interesting data that looks at metabolism and reactive oxygen species. So I think there's a certain threshold that once you get to that threshold, then the cell starts dying. And sometimes you can get faster to that threshold if you use some of these drugs in combination, so lenalidomide plus a proteasome inhibitor. And having the ability to test for that will be critical because as we are discussing triplets, and if you're going to start a patient, say, with carfilzomib, Lendex, a patient who has refractory multiple myeloma, and the patient responds, are they responding because of the carfilzomib? Is it the Len? Is it both? But you could easily envision that someone responds and it's only the carfilzomib that is providing that benefit. So I think our ability to test for that is really, really mandatory at this point. We don't have good markers. My group and others are working towards the development and validation of clinical biomarkers that would allow you to do this prediction. But as of now, it's a bit of a guessing game, and we're just extrapolating from large data sets to say everyone should get a triplet. However, because we don't have those markers, it's not uncommon that we switch from lenalidomide to pomalidomide when a patient has the suspicion or we have the idea that the patient has been refractory to lenalidomide on the basis of our clinical observations alone. There is no question when you look at the laboratory data and all the studies that have been done in vitro, pomalidomide is a more powerful inhibitor of the pathways that we think are critical for cell death.
You know, the thought process is that if you've been on an IMiD before, you need to change from one IMiD to another. And I don't think that's necessarily true. I think going from IMiD 1 to IMiD 2, there's adequate data now in the POM space where most of these patients have been Len refractory, and in these Len refractory patients, you're still able to capture responses with pomalidomide, whether it's with pomalidomide and bortezomib, whether it's with pomalidomide and daratumumab. It's an open question, and you can use either or carfilzomib, for example. So you can use one IMiD after another. You can use one proteasome inhibitor after another as well. And it really speaks to the potency of these drugs. So even if you've had bortezomib early, you can be rescued with carfilzomib because carfilzomib is a fairly potent irreversible proteasome inhibitor. So you can use the same class of compounds as long as it's a different compound.
The fortunate news in myeloma is that we actually have second-generation immunomodulatory drug pomalidomide and second-generation proteasome inhibitors, carfilzomib and ixazomib. And those agents actually were FDA approved because clinical trials showed they work even when the first generations don't. So I've mentioned that if you have the three-drug regimen, lenalidomide, bortezomib, dexamethasone, we can switch to pomalidomide dex with either a second-generation proteasome inhibitor, carfilzomib or ixazomib, or a monoclonal antibody, daratumumab or elotuzumab. If it's unusual, but if you're treating with a doublet, so you were doing lenalidomide, dexamethasone, for example, and the disease grew, then we would switch to the opposite class of drugs. So we would switch, if the patient hadn't had a proteasome inhibitor, we would then start the same. The converse is also true. If a patient, for example, had had Velcade dexamethasone initially and the disease relapsed, then we would switch to an immunomodulatory drug. So that's the primary setting in which we might switch there. And switch also might mean, when do we first use antibodies? And that's adding in daratumumab or elotuzumab to the pomalidomide dex. So that's really a switch, since they haven't had monoclonal antibodies before.
So carfilzomib, is something which we are still trying to figure out the best dosing and the best schedule. There's no question in my mind that it's a very potent proteasome inhibitor. I think doing it twice a week for 3 weeks is going to be retired. I do think it's going to be used as a weekly schedule. Given that we have been able to go up on the dose of carfilzomib, so we started off at 20 and 27, and we're now using 70 mg per meter square, and the 70 mg per meter square given once a week is actually well tolerated. I think the future of how carfilzomib is going to be used is on a weekly schedule. Now, what dose you use on a weekly schedule would largely depend on what the partner drug is. I tend to use carfilzomib more with an IMiD combination, and I will say that when you use it with an IMiD combination, I don't think you can go up beyond 56 mg per meter square. 70 has been tried. We have used it, but typically most patients do well with the 56 mg per meter square with the IMiD because of the toxicities of cytopenias associated. So using carfilzomib on a weekly schedule, depending on what your partner drug is, would tell you what the ideal dose of carfilzomib should be. I do think even in the upfront setting, carfilzomib should be used on a weekly schedule. It's extremely well tolerated and combines well with most of the drugs. And there's data now at ASCO from last year with carfilzomib was used with daratumumab, for example. Carfilzomib was used with isatuximab as well on a weekly schedule, and extremely well tolerated, going all the way up to 70 mg per meter square.
The benefit of weekly carfilzomib is both to the caregiver and the patient. Up until now, we've utilized carfilzomib twice a week for 3 weeks in a row with 1 week off, because that's the original schedule and regimen that was FDA approved. But more recent data suggests that you can safely use single doses of carfilzomib and even have escalated or higher doses and use this very safely. So this honestly gets equal efficacy. That's very important, and is much more user-friendly in terms of clinic time and in terms of times, that patients have to come in to visit us in the clinic for treatment.